Chapter 9, the Immune System, Screencast 2. Now, this screencast is focusing on barriers and the first line of defense. So if we look at this list, so skin, incredibly effective until you cut yourself and then you allow things in. Within our tears and saliva, they actually have lysozymes. And what a lysozyme is, that's an actual antibacterial enzyme. If we look at earwax and mucus, both of those, think of them as sticky, will be able to trap microorganisms. The stomach has an incredibly highly acidic environment, and that will actually inhibit microorganisms. If we look at the vagina, that also is slightly acidic and inhibits some microorganisms. Vomiting, urination, and defecation, those are processes responsible for the removal of microorganisms. And then we have resident bacteria, and they will actually outcompete pathogens. Now, if we look at these non-specific defenses, so this counts as a second line, we've got phagocytic cells. These are white blood cells, and what they do is they surround and engulf invading bacteria. Typically, if we look at neutrophils, macrophages, and eosinophils. The inflammatory response, and if we look at signs of the inflammatory response, redness, warmth, swelling, and pain. There are cells known as natural killer cells. They are a lymphocyte that attacks tumor cells and those cells that are infected by viruses. Complement proteins, these are plasma proteins that actually invade bacteria when activated. Interferons are specific to antiviral proteins, and then fever. So these are all non-specific. They can be brought about by various types of things, but note some work better with others. For example, complement primarily are most effective with bacteria. Interferon is most effective against proteins. Here is just an overall chart of the primary second lines of defense. I would recommend taking a look at this because I think it really is helpful and it puts everything in one place. Here, this is actually showing the activation of complement. Think of complement as kind of tagging a protein. That's really what it does. And so one of the things that might happen is that complement will be responsible for creating holes in the wall of the bacteria. Now, because there's holes within the bacteria, water and salts can diffuse into that bacterium through the holes, and then that bacteria will actually swell and burst. And you can see that here as far as the micrograph. Okay, and this process is called lysis. Okay, so it's been actually activated by a complement protein. Here we're looking at the process of phagocytosis. I always kind of describe this as Pac-Man, gobbling things up. So here we have the phagocytic cell, and notice what happens. The bacterium forms an invagination, and then the cell membrane closes behind it, and now it's a vesicle. And now you've got the bacteria inside the vesicle. Then the vesicle will fuse with lysosomes. And remember, these are organelles that contain digestive enzymes. The enzymes will break down and digest the bacteria, and then what's left will be excised through exocytosis. Here we can look at the general process of as far as damaged cells and mast cells. So here's the inflammatory response. And so what we're looking at here, histamine and other substances, they're going to dilate blood vessels and they're going to make them leaky. That accounts for swelling. The complement protein from plasma, they diffuse out of leaky capillaries. They mark the bacteria for destruction and may kill them themselves. And then, attracted by histamine and other chemicals, phagocytes squeeze through the leaky capillary walls and they begin attacking and engulfing bacteria and debris.